Step back into the 17th century to discover the remarkable legacy of Maria Sibylla Merian. As a single mother and intrepid explorer, Merian fiercely defied the rigid scientific dogma of her era. This slide introduces her transformative journey, illustrating how she fused breathtaking artistry with rigorous empirical observation to unveil the hidden, interconnected lives of insects. By masterfully documenting these relationships, symbolized here by the vibrant Dido longwing butterfly perched upon a pineapple, she did far more than just paint nature. She revolutionized our understanding of the natural world, ultimately giving birth to the foundation of modern ecology. To truly appreciate Maria Sibylla Merian's impact, we must look at the stark contrast between the status quo of her time and her revolutionary approach. On the left, we see the 17th century status quo. Science was dominated by wealthy men studying dead, isolated, and dried specimens pinned inside indoor cabinets. The prevailing dogma even claimed insects were beasts of the devil, born spontaneously from mud and rotting meat. On the right, the Merian method completely shatters this paradigm. Merian proved that insects actually hatch from eggs and undergo predictable, beautifully documented life cycles. Her core philosophy? Insects must be studied alive, observed directly in their natural habitats, and understood in relation to their specific environments and food sources. Let us trace the roots of observation that shaped this pioneering scientist. Born in 1647 in Frankfurt to a renowned Swiss-German publishing dynasty, Maria Sibylla Merian's early environment fueled her creative intellect. From 1651 onward, she trained under her still-life painter stepfather. Though systemic guild restrictions barred women from painting in oil, Merian adapted brilliantly, mastering watercolor, gouache, and copperplate etching on vellum. By 1660, at just 13 years old, her scientific curiosity truly ignited. She began rearing silkworms, meticulously keeping detailed empirical logs of their incredible transformations. These early milestones, combining artistic mastery with disciplined observation, laid the crucial foundation for her groundbreaking ecological discoveries. We now enter the domestic laboratory, exploring the meticulous workflow that drove Merian's research. First, she established her garden collection. To support her family, she formed the Jungfern Company, teaching drawing to wealthy, unmarried Nuremberg women. This granted her exclusive access to their elite private gardens for specimen collection. Next came the relentless rearing inside custom wooden breeding boxes. Merian raised hundreds of species simultaneously, tracking larval growth, pupation, and emergence with unmatched precision. Finally, we see her late-night illustration process. Through dedicated empirical documentation, she famously stayed awake all night by candlelight, using watercolors to capture the exact, fleeting moments of insect metamorphosis as they unfolded. Here we witness a monumental shift in scientific history, dismantling spontaneous generation. With the landmark publication of Der Rapin, the Caterpillar Book, in 1679, Merian provided undeniable, sequential visual evidence that completely dismantled the long-held Aristotelian belief in spontaneous generation. Instead of relying on philosophical speculation, she presented a clear, cyclical journey of life, from egg to larva, passing through multiple instars, transitioning into a protected pupa, and finally emerging as a fully formed adult. Merian didn't just write theories about insect metamorphosis. She fundamentally proved its existence to the world through rigorous, empirical visual data, cementing her place as a true pioneer of life science. Let us examine the anatomy of an ecological masterpiece, where Merian's artwork reveals three core ecological insights that were centuries ahead of her time. First, she documented host plant specificity, proving that specific larvae will die if they are not fed their distinct, native host plant. Second, she unlocked early concepts of parasitoid biology. Merian identified the mysterious reason why some caterpillars yield wasps instead of moths. Parasitic wasps lay their eggs directly inside the living larval host. Finally, she illustrated phenotypic plasticity, showing how a caterpillar's growth and coloration can vary distinctly based on environmental factors and diet. Through this single, integrated drawing, Merian masterfully captured the complex, real-time dynamics of an ecosystem. This slide captures a pivotal turning point driven by profound frustration. After leaving an unhappy marriage in 1685, Merian settled in Amsterdam by 1691, establishing a highly successful, female-led workshop. Through her success, she gained exclusive access to the prized specimen cabinets of wealthy merchants. However, staring at the dead, pinned exotic creatures, she realized they told her absolutely nothing about their living ecology. In her own words, encountering these static collections where origin and reproduction is unknown, only begged the question of how they truly transform. Driven by an insatiable need to see these creatures alive, she resolved to leave Europe behind and undertake a long-dreamed-of, daring scientific journey to Suriname. We now arrive at a historic milestone, the 1699 Suriname expedition, a journey that defied every social convention of the era. First, consider her circumstances. At age 52, Merian departed Amsterdam completely unaccompanied by any male sponsor, traveling across the Atlantic only with her 21-year-old daughter, Dorothea Maria. Second, the voyage was entirely self-funded. Receiving absolutely no royal patronage or commercial backing, she paid for the perilous 5,000-mile journey to Paramaribo independently by selling 255 of her own paintings. Their singular mission? To study and document the entire life cycles of tropical insects directly within their native South American rainforest habitat, marking one of the earliest purely scientific expeditions in history. Let us step into Merian's reality during her intense fieldwork in the jungle, which reveals both her progressive scientific mindset and the realities of colonial Suriname. First, she was active in rejecting monoculture. Merian openly and boldly criticized the Dutch colonists' myopic obsession with cash crop sugar exports, choosing instead to focus her lens on documenting the region's vast, untapped biodiversity. Second, her breakthrough findings required vital collaboration. As she trekked deep into the grueling, humid rainforests, her success relied heavily on the profound ethnobotanical knowledge, physical labor, and guidance of enslaved Africans and indigenous Amerindians. Without their indispensable expertise, she could never have located or truly understood these exotic species in their natural environment. We now behold her crowning achievement, the publication of Metamorphosis Insectorum Surinamensium, 1705. This monumental work featured 60 copperplates, etched meticulously from her precise watercolor field notes. To ensure maximum accuracy, she utilized expensive, delicate hand-colored counterproofs to replicate the exact softness and vibrant hues of her original artwork. Remarkably, the masterpiece was entirely self-published, financed independently through advance subscriptions from wealthy collectors. The ultimate impact of this volume was profound. As the first-ever natural history book completely devoted to Suriname, it utterly shocked Europe with its raw, vibrant, and complex depictions of tropical ecosystems, solidifying Maria Sibylla Merian's legacy as a foundational figure in both science and art. We conclude by visualizing the food web, revealing how Merian acted as a Darwin before Darwin. Her plates rejected simple classification, instead, they captured the brutal, interconnected struggle for existence. By vividly illustrating food chains, multi-species predation, and devastating ecosystem collapse, such as army ants stripping trees bare, she pioneered the documentation of behavioral ecology more than a century before the field was named. The artwork masterfully visualizes these trophic levels in a single snapshot. A leaf acting as the primary producer, a hummingbird feeding as the primary consumer, and a massive spider towering above as the shocking apex predator. Through this raw lens, Merian proved that nature's true beauty lies within its complex, unyielding relationships. We now turn to a deeply moving chapter of Merian's work, Ethnobotany and the Peacock Flower. Here, her observations transcend entomology to document a profound human tragedy. Merian recorded that enslaved Amerindian and African women utilized the seeds of the vibrant peacock flower as a botanical abortifacient, bravely refusing to let their children be born into the horrors of Dutch slavery. Because this crucial data was entirely excluded from traditional European pharmacopoeias, Merian's record serves as a rare, vital preservation of indigenous medical knowledge and marginalized resistance. It stands as a powerful testament to her holistic approach, capturing both the natural world and the painful human realities embedded within it. Let us step into the matrilineal workshop, a remarkable multi-generational engine of science and art. This slide maps the collaborative four-step pipeline that powered the production and global distribution of Merian's masterworks. It begins with Maria Sibylla's primary field notes and watercolors, which her daughters, Johanna Helena and Dorothea Maria, translated through intensive technical labor into copperplate etchings. Following the studio's signature hand-colored counterproof finish, these masterpieces reached elite international collectors, including Tsar Peter the Great. Crucially, these daughters were master naturalists in their own right. When a stroke paralyzed Merian in 1715, it was their enduring expertise that ensured her legacy was successfully published, preserved, and distributed across Europe. We conclude by examining erasure versus revival, tracing the tumultuous historical legacy of Maria Sibylla Merian. During the 19th century erasure, Victorian-era male scientists dismissed Merian as a mere flower painter. Critics like Lansdowne Gilding aggressively discredited her observations, such as mocking her depiction of a bird-eating spider, and weaponized her reliance on the knowledge of indigenous and enslaved peoples to diminish her authority. Fortunately, history corrected course with the modern revival. Modern biology has fully validated her once impossible claims, and her early ecological frameworks are now celebrated as foundational. Today, her legacy is beautifully cemented as newly discovered species, like the vibrant butterfly Catastocta sibyllae, are proudly named in her honor. We reach the final summation of our journey, the core insights of Maria Sibylla Merian's profound legacy. This slide beautifully synthesizes her revolutionary impact across three key dimensions. First, we honor her as a pioneer of ecology, the visionary who shifted biology away from static, dead specimens toward the dynamic study of living interactions and life cycles. Second, her work exemplifies art as empirical data, proving that rigorous illustration can serve as precise scientific proof. Finally, her story is a triumph of intersectionality, showcasing how a woman independently carved a path through colonial and gender barriers to reshape science. Merian's life reminds us that true discovery requires us to see nature not as isolated objects, but as a deeply connected whole.